Start a transect at this speed. This is fine.
All right, speedy transect. This is T7.
And yes, Liz, Thesia is on the collections list if we see it.
Whoopsie.
Well, they also could have been Asbestopluma.
Okay.
All right. Roger.
You've seen snakehead eel pouts, lots of sea stars and sea anemones. I think these are the branching Asbestopluma on these rocks.
Got a line here.
Oh, do you want me to stop? Let's see.
Stand by. Looks good.
Looks like some derelict
Yeah.
line, possibly fishing gear. But luckily it's not up in the water column, it looks like. It's pretty well flat on the bottom. It's one nice thing about having Argus up there to get a view of that.
Some plexurid corals just went through.
Yeah.
We're good.
We're getting a few more little rocks in here, so we're seeing a few more corals because they have something to hang on to.
That one looks like a bamboo.
Science, I'm going to change my heading a little bit so that I don't get blown off by the current all this time.
Copy that.
More kelp fall. Little barrel sponge out there. I can't even see the rock it's clinging to, but I'm sure there's one under there.
I think these branching ones are all Asbestipluma.
Anyone recenter on there, please?
Yep.
And they sampled one earlier, but I don't know if it was a branching one or a single stick.
When we're done with this transect, Meredith, give us a shout if you want one of these branching Asbestipluma, and we'll look around and grab one for you if you do.
Oh, Abby's on now, so maybe she can answer the question.
And Meredith's on.
Yeah, we're going to end the transect about 90 meters from here, so
Roger.
Okay.
hopefully this stays asbestos plumacy.
So Di's saying it was a single stick, so yes, we would probably like to sample one of these branching ones as soon as we finish with the transect.
Roger that.
Oh, you're talking about the carnivorous sponges? Is that the signals?
Correct.
Oh, okay, cool. Yeah, I wonder what's going on geologically here as we get up this other side of the bank. There's a little kind of bump. It looks like it's getting a little more rocky. A little bit. It's a little bit more carnivorous spongy.
I liked your Asbestipluma-y.
Yeah. Not sure the sponge experts will. Are they the one genus of carnivorous sponges, or are there several?
I've heard of at least two. I think there's a Cladorhiza. I know there's Cladorhizidae as the family, but I think there's also a species or a genus of
Roger.
Cladorhizidae, Cladorhiza or something. How do they end? How do genus end? Usually.
AE, I don't know.
Genera. Yeah.
DAE. No, that's family. I don't know.
That's family, yeah.
Yeah. Right, let's see. Another 50 meters.
Roger. Sorry?
Yeah.
Yeah.
We'll probably come back down a bit too.
Sorry? Yeah, a little more
So
rocky.
when we talk about carnivorous sponges, it's not like they are moving around and capturing prey, but instead of most sponges really strain bacteria and bits of organic material from the seawater, carnivorous sponges have little hooks that capture tiny animals that are swept through, little plankton that are swept through. So they have these Velcro-like hooks that ensnare prey. So that's why they're called carnivorous. And they have really odd shapes. There's one that looks like Erica was talking about those chondrocladia sponges. There's some that look like harps, and some that actually look like have little stick-like processes with look like ping pong balls on the ends of them.
So this is 200 meters, and we can mark the end.
Roger that.
So is that the harp sponge they found in Monterey Bay a couple years ago? I remember hearing about that, like lyra, something.
Yeah.
Oh, that's
yeah.
carnivorous. I didn't even.
Yeah, it's chondrocladia lyra.
Yeah,
it's so pretty.
Yeah, I remember the photo.
They have these harp-like
So science, you
processes
wanted.
that stick out. Yeah, let's look around for one of those.
Did you want one of those sponges?
Yes, it'd be great.
All right, Ben, you want to stop the ship and we
yeah,
can
we're
look
done
around?
with the move.
Roger. Or kelp.
And of course, now I don't see any. Well, that's kind of a bummer.
Looks like there might be one viewed here.
Oh, these little ones. There we go.
We'll wait for that ship move to kind of settle out.
Yeah, I can bring us back to.
I think you're good, Ben. I think you don't have to bring us back anywhere.
So Abby is saying that most of these Vesipluma aren't branched, but there are some that are branched.
Roger.
Can you still hear me? I just got knocked off the link.
Oh, yes, sorry. Yes, we still hear you.
Okay.
About right below the whip coral, is that?
I think we're just about
Barrel
settled out.
sponge has kind of a yellow cast to it.
Abby's saying that there are about 130 different carnivorous sponges.
Oh,
Wow.
Wow.
so their spicules are like modified from non-carnivorous sponges. Wait, that's another Abby question.
Correct.
Yeah.
Yeah, no, that's right. They are.
All right, science, let me know if you see a sponge that you want. And for sticks.
Will do.
See something on the left.
Those over there, do you think?
We've gotten one of the single sticks, so we need
Oh,
one that
the
has
branch.
multiple sticks.
Right.
Yeah.
Oh, Raj.
Four-leaf clover.
It's
And they were
guaranteed that
super
every time we
plentiful
start,
in the mud, and now we're back in the rocks. And
Yeah.
they faded out.
every time I start a transect, we go over something we wanted. You
I
can almost
goaded you
guarantee
into
it.
it, though. My fault. Let's see. Looks like north of here is a little harder than south. So, yeah, I don't know. This would be softer. This would be harder.
Sorry?
The backscatter. This is softer.
Yeah, so if
Mine
we go a
is
little
super
south.
coarse, yeah. Again, very coarse, but
So Liz, do you want to backtrack, or do you want to keep moving forward, or stay
I don't
here on the
know.
ground,
I would tend to backtrack, quite honestly, but I'm not sure whether that's doable for the ship to do that kind of move and the ROV to turn around and go back to the sandy area or not.
yeah, it's not a problem for us. It's just a matter of it will just take a little bit of time.
It'll just take away from going the upslope. We can also maybe go a little south, like Ben was saying. There's some softer targets and backscatter potentially,
Yeah, but
that's not a guarantee.
Why don't we turn around and
Roger.
I know it'll take some time, but
Well, I mean, I know they want to get to the slope, so maybe we should ask Abby if this is one that we need.
Okay, Abby, is this one you need?
And again, once we get from to 3F to 3G, that's about 45 minutes at 0.3 knots. And we're probably going to get off bottom at 11 a.m. around. Maybe a little.
Let's do the math.
Yeah.
Yeah, we're just running short of dive time, Liz. I don't mean to.
Yeah, I'm trying to see if Abby's going to answer us back.
Yeah, so we'll probably want to be off bottom at 10:45
Okay.
local time. So, and then
Well, why don't no,
Abby's saying
continue up
carry
slope.
on.
Yeah,
okay.
carry on. But if we can ooch a little south, like you said, and see if we can go over a little bit more sand, maybe that's the in-between
Let's try that.
choice.
Roger.
I'm going to look at that. Yeah, let's go just 50 meters south.
Okay.
Is that good? And then we can go to the target from there.
Roger.
Yeah, I mean,
Sorry?
nothing. Just,
Oh yeah, go ahead. Maybe you wanna, yeah. Ben, can you show me where, did you mark a target when that fishing gear was? Or that line was?
no, I didn't. I'm sorry.
Raj. Kim, you wanna go due south?
I can do a nominal target based off you changing our heading, but I don't know if that's. I'm thinking it's 160 meters or more behind us.
You wanna stop that ship move for a minute there, Ben? I'm gonna turn to the heading and then back up a little bit to get us some slack in the tether there, Kim. Raj. Actually, Ben, I'm pretty south, so you can actually start that ship move.
All right, let's just do 40 meters and
Yep, sounds
go
good.
from there.
All right. Yeah, we'll just wait for the ship move. Yeah. Looks like you'll just have to look up a bit there, Kim. Not making any lateral motion though.
Yeah, we've come 20 meters south. I don't know how important it is to.
Oh, I see something branching there.
Oh yeah. Oh yeah.
Uh,
Science, is this a potential field of interest here?
That branching.
I only see the one branching, and it's only a single branch. The ones we were seeing earlier had multiple. So if we can hold out for a multi-branch.
So that one there, that pitchfork one is, we want multi-branch, Roger. Roger. All right. expect another ship move there, Ben?
Yeah, let's start heading back to the target after this. Is that good? 40 meters south is what we've come after this ship move.
Sure.
And then, is that good? I mean, there's still a chance there'll be the Asbestipluma.
Are we over that soft target there?
Yeah, let's see.
Yeah.
Yeah, oh wait, let me switch the meters.
Yeah, we can just go back to the target after this.
Yeah, so after this move, I mean, just 20 meters south, you're in the 4 pixel zone.
Raj.
So,
Interpolated.
yeah. But it definitely looks a square of softer stuff. So it might bring us over softer stuff as we go to 3F.
Roger that.
It's all interpretation of a proxy.
We'll keep looking.
Yeah. Jess wants to get it. I want to get it. Yeah, and there'll definitely be time to sample and play around on that slope. I think it won't be more than an hour and 30, 40 minutes to get all the way, do all that. Get there, go up.
I don't know. Abby's coming back on shift. You might
Oh, right.
be collecting a lot. I see another 2-brancher, but
Yeah.
It's maybe finer sediments too than what we were seeing back there.
Could be.
Because the other returns weren't so muted. We're done with the ship move if you want to start heading back.
Roger that. We have the
Good.
target bearing.
Yeah, it's going to be 285 now.
285. You want to stand by on that while I get in position?
Perfect. Yeah.
some of the Death Star sponges, and I saw some Paragorgia coral. Baria sponge. Some more of the Thanea.
All right, Kim, I'm going to just drive under you and follow the DVL screen, I think. Too far.
Do you want to reset or do you not want to change the trail?
Stand by.
Yeah. We're back here.
Roger.
So Abby is saying that if we see a 2-brancher and that's all we can get, we should get a single brancher as well that's nearby, so she can make sure they're different species.
Roger. It looks like there was a 2-brancher on the rock that we just came back from. Did you want me to go back there?
No, let's just proceed. We'll hopefully see another 2-brancher.
Roger that. But now that we say that, we're going to see a 4-brancher.
Then we're definitely getting that one.
Oh, yeah, for sure.
Yeah.
All right, Kim, if you want to do your spin while we still have some tether. I'll do 285, correct?
285. Just let me know.
Stand by.
What is that?
All right, Ben, you want to reset the DVL?
Yeah.
Thank you. And then I'm going to do a little wrap mitigation here.
Yep.
Let me scoot over a little bit before I do.
You're hearing the complicated dance that has to be done between both ROVs and the ship. It's even more complicated than it sounds.
Mm-hmm. Actually, Kim,
All right, you ready?
yeah, let's do it.
Okay. Good.
Roger. And Kim, would you mind coming down on the delta a bit?
Okay. We did the asbestoplumus dance.
Yeah, well, we tried.
A for effort.
Mm-hmm. Yeah, yeah, until we get to maybe the contour at the bottom of the feature. That'll probably be the last move of our shift, actually.
Yeah, I think so. I'll tell Sam to keep the pace a bit. current getting stronger on the side?
Yeah, yeah, it's been pretty strong the whole watch, I think. I've been going to like 40% lateral thrust to maintain position. That's why I've been just turning into it like that.
Yeah.
So I don't use my lats so much.
Let another 30 for them.
All right, guys, video is switching over.
So we are still on Santa Lucia Bank. We're heading over to a steep slope that we're hoping to climb. And we're also in the middle of a shift change here.
Erica, I'm going to drop off and give you time to brief, I guess, Lizzie when she gets on.
Okie doke.
Okay.
All right. Thanks, Liz.
All right. Thank you.
ROV changing over. Thanks, guys.
Looks like we're back into the abstract sponge section. Still seeing some bamboos, maybe some asbestipluma, mushroom coral there.
Test, test.
I hear you, but you've got an echo.
Oops, I will fix that right now. There we go. Good morning.
Hello.
Hello, how was the 4 to 8?
It was fun. We had some slopey stuff in the beginning, and then we switched over to the transit across the flat, and I think we've got you guys ready to climb up another slope pretty soon.
Oh, how kind of you. Thank you. That sounds awesome.
Yeah, so I think Guy is back on at this point, and he was the one kind of giving directions as to the areas he was hoping to take a look at. So this 3F point was one that he wanted to climb up the steepest point.
Okay.
To get to that.
I see a little paracordia.
Yeah, we were seeing quite a few corals on the steep, so hopefully you guys will see the same.
Yeah. I'm pulling up the dive plan and the dive data recovery report, but I saw you and Abby were chatting about a couple sponges that they might have collected so far.
Well, we found some branched asbestipluma on our last transect, but we ran out of them right at the end of the transect, so we weren't able to get one. So keep an eye out for those.
Okay.
And we collected a lumpy barrel IKEA lamp.
Good morning, Shoreside.
Hello.
H12 is ready to go in the van, and it looks like we are still heading towards waypoint 3F, correct?
Yes, from what I understand, yes.
Roger.
All right,
Nav.
I am going to switch out if Abby's ready to come on, and have a great shift.
Thanks, Erica. Good morning, Abby. No, you sound good.
Yeah, go for it.
We do not hear Abby this morning. Heard you dive, but we didn't hear Abby.
Oh, this is Lizzie. I've actually.
Oh,
There she is.
I was tricked. It was doing the thing where the scientists are sure we can hear each other even when the talk bars aren't clicked.
Okay.
So this little patch of yellow here that we're just about to sail over would be something to have a little look at.
Is that something that we need to zoom or
If
close
we
up
could
on?
get a zoom, yeah, it's just under that heteropolypus. So there's a few polymastia species that, well, we think it's polymastia, but I've been seeing them and I'm not so sure. But this is definitely something that stands out on the
All right, Al, let's zoom
AUV
in.
imagery and on the ROV feeds. And to my knowledge, we
There
do
you zoomed.
not know
Yeah, I'm
what
coming
this
back.
is. Oh no, so it's more of that.
Oh, let's come wide.
Yeah.
The yellow eggy mast.
Yes, yep. Tricked.
We got some of that last night.
Okay
Yep,
to continue
yep,
on?
we did. Yep.
Oh, and Finea, lower left.
Yeah, Bob was also able to collect an excellent sample of that Finea last night. It was just perfectly on a cobble.
Very nice. It's so great when you see something perfectly perched to sample.
There was a selection process involved.
So there's been a little bit of a schedule change. My name's Lizzie Duncan, and I work for the Channel Islands National Marine Sanctuary, and I'm partnering with Abby for the 8 to 12 for the next couple of days. Previously, Abby was working with the fabulous Di Water, so apologies if you were expecting her today. Big shoes to fill.
Lizzie, what is your sign-in username?
My sign-in?
Yeah. The username.
The Lizzie
Sigh.
Duncan?
No, the sign on
Oh.
D-Link.
Oh, lead A, I believe.
Lead A. All right, thank you.
I thought you were talking about the science chat, but I understand now. And Abby, do you want to introduce yourself as well?
Sure, I'm Abby Powell. I work as a contractor at the Northwest Fisheries Science Center up in Seattle with the AUV group that's run by Liz Clark up there. And my background is sponge ecology, and I have been keeping track of the sponges that we've been collecting on this and previous dives in the Channel Islands and at Santa Lucia Bank.
Awesome. And today we're diving near the Santa Lucia Bank just northwest of Point Conception, and we're at roughly 950 or so meters. And we're looking for sponges and corals and all the other cool things. This is an area that hasn't really been well explored before, so we're the first discovering what kind of habitat and seafloor is looking like. And I know from our first dive, it was a lot of unconsolidated cobble, but I've seen a bit of both. I've seen consolidated bedrock on the previous shift, and now we're seeing a little bit of cobble. So looks like we're getting more towards the habitat we were hoping for.
Yep, so we'll have explored two
nice big sponge.
slopes with different aspects. Oh yeah, big glass sponge coming up here. So the previous shift traversed from the one slope across the sort of flatter cobbly area, and now we're just arriving at a slope on the other side of this sort of valley feature.
And it looks like the last shift set us up really nicely to head upslope, which is usually pretty exciting because the steeper the slope, typically it means the less sediment has a chance to accumulate. So we're hopefully going to see more hard bottom like we're seeing now. And I do believe our priorities for this dive are more exploratory than they are necessarily quantitative, which we usually, if it's going to be quantitative, take some transects, which I know we have a few. But I think the priority is to cover as much ground as possible, and then while we're en route, if we're going the right speed, to take a transect. Oh, is that a nice?
All right, Al, let's get a quick zoom.
Is that a ray? I think that's a ray.
I think that's a rough-tail skate.
Do you know, Abby?
I think it's a rough-tail skate. See those little spines all along its tail there.
We also have a wonderful science chat. Rough tail skate. Thanks, Tom Leidig. I knew someone would be in there with the right answer. Which maybe the giveaway are the spines on the fins and the tail there.
Okay, let's come live.
Lizzie, can you hear Abby? Lizzie, can you hear us on the ship?
Uh-oh.
What was our new heading, by the way, Sam?
Same heading, 287.
I second that, uh-oh. So again, someone on the chat, let her know, please.
287, got it.
Paul, I gave you a junk zoom even though you didn't
Yeah,
ask.
perfect, thank you.
I did that thing again where I didn't know no one. I couldn't hear anyone else and I just kept talking. Sorry, guys.
Can you hear us
That
now?
happened to me the other night. Yes. Hopefully I wasn't talking over anyone. I was just trying to fill space. I wonder why that happens. It doesn't really give you a clue. It's not like anything's muted or error looking.
Can you hear us now?
Yes, thankfully.
Okay, good stuff. I was just like, rough-tail skate, rough-tail skate.
This is part of the technology which amazingly brings both Abby and I to the Nautilus and to the public, is this V-Link software. But this is, I believe, OET's first time using it and being in two very different places and everything, you know, satellite, and this has to travel really far to the satellite and then back to the Nautilus and to all of you. So there will be some challenges along the way, but it's a pretty amazing technology. So you're in Seattle right now, right, Abby?
Yeah. Yeah, no, I think on the whole it's been amazing.
Yes. And then I'm in Santa Barbara. So we are not on the ship, but we get to join the control van with Nav, the ROV pilot, video, and our data logger in the comfort of our homes and or offices.
Except food doesn't appear at regular times.
All right, Al, let's zoom in.
Nice little lysocene glass sponge. We know that there are a few species and genus that all look fairly similar to this. We're still trying to unravel who's who.
I see a little branching coral there that just went off down to the right. And are these long white sticks, are these Asbestopluma or are these something else?
I think those are sea pens.
Oh, see, oh, okay.
Meredith is still on, so you should take advantage if we see some corals to.
Can we look at one of those the white whips again? Because I think they're attached to the rock, which might not make it a sea pen.
Oh, yeah.
Just want to be sure.
Yeah. Yeah. I was wondering, like, well, they might have found the very smallest bit of soft sediment then.
Mm-hmm.
It's thrifty. So Meredith Everett also works with the Northwest Fisheries Science Center, and she is a geneticist who primarily works on corals, but she does genetic analyses for a number of organisms.
All right, Al, let's
and
zoom in.
she's in the science chat with us, but she's in the lab today, so she'll be in and out offering her IDs.
Is that an Avesta pluma in the bottom right or bottom left?
On the...
Yes, on the bottom left. Oh, that's quite a nice shot because you can see both side by side.
Good lasers. Can we go tighter?
So the little pipe cleaner on the
What
left
was that,
is a
Steve?
carnivorous
Go tighter?
sponge.
On that
Yeah,
white.
can we go zoom in now?
Yeah, you're going to have to tilt up to do that. Otherwise, it'll be a mess. Well, not that much.
Yeah.
No, I know how the sled works. Bump up just a little bit, I can go all the way in.
Is that all we got?
You got to tilt up.
Can we get
more?
closer?
I think it's, yeah, I can get closer. We don't have a lot of time, but yeah, let's come wide.
Okay.
Science, is there interest in staying here longer or taking a sample?
I'm not sure